And before I forget, I'd just like to thank our panelists. Building apps and really doing large scale analysis on the RAP is something I get asked about literally every day, even on the weekends, because somehow people find my contact information on the internet. So I really appreciate these people coming and really talking about their experiences. This is an experiment my colleague Brenton and I have been doing over the past few months is really allowing UKB-RAP users to talk about their experiences using the UKB-RAP. We find that it really helps people. We did one of these on epidemiology, I believe 3 weeks ago, maybe a month ago. Please, if you're an epidemiologist, please feel free to check out that one. I think it was really great. So fantastic. So a few housekeeping announcements before I introduce the panelists. Really, the easiest way to get your question through to us is with the Q&A module at the bottom of your screen. So if you have questions for any of the panelists, please go ahead, put them in the Q&A module, and then I will read those questions to the panelists towards the end. That should be really cool. Also, on a personal note, if you are going to the UKB Scientific Conference in London on December 10th, please feel free to reach out to me individually. We have some satellite things going on there. Love to see you all. Love to talk to you about the RAP, et cetera, et cetera. So again, if you're going to the UKB conference, find me, Ben Busby, and I can clue you in on some of the other things DNA Nexus is doing around that conference that we're really excited about. All right. So with that, Brenton, have I hit all of the housekeeping announcements?
Just wanted to mention up top, additionally, this webinar is a bit more of an advanced topic. So if you do not have command line experience, I can point you to some good materials here on our Academy documentation, where you can get that background info. But for this webinar, some familiarity will be required.
Yep. And if you're not there in a career sense and you want to be there, I'd highly recommend the carpentries, particularly software carpentry. That'll get you started on your sort of command line journey. And at least in my experience and the experience of most of my collaborators, will improve your life overall. So basically, learn to code. It'll change your life. So with that, let me introduce you to three people who can code. So we have three speakers today. In order of appearance, there's Emily Page, who works with me at DNA Nexus. And years ago, or at least over a year ago, I convinced her to come to DNA Nexus, a fact of which I am very proud. And then Xing Chan from Aidan Doherty's group at the University of Oxford. Hopefully he'll be around for the scientific conference, and you may get to meet him. Really, they're doing some amazing stuff there with multimodal data. Finally, Phil Greer, friend and collaborator who I chat with and work with quite a bit. We have a lot of common interests in Hardy-Weinberg equilibrium, as well as many other things. So I'm really excited he's here to talk to you. And many of you, I know, have been in the sort of UK Biobank larger community for a while. And so many of you have probably used some of the tools that Phil has put on GitHub over the years for GWAS and other things, and certainly was a huge contributor to the DNA Nexus UKB community when that was up. And if it's not up anymore, but it's been archived, you can check out things, but you can also go to the UKB community itself and check things out. That said, I think we're through all the introductions, introductory announcements, etc. So I'm going to turn the floor over to Emily to go ahead and sort of give an introduction into building apps and workflows, and then we'll go to Xing and then Phil. So without further ado, here's Emily.
Great. Hi, everybody. My name is Emily. I am the Academy instructor here at DNA Nexus. So my job here is to help teach you how to build apps and anything else really you're wanting to do, both on the core platform and also within UK Biobank. So let's get started. All right, let's first start talking about the differences between apps, applets, and workflows as you're getting familiar with these topics. So apps and applets are both ways for you to execute code. The biggest difference between them are their visibility. So apps are what you see in your tool library. Applets are what you build, okay? Then you have native workflows. Native workflows are built within the UI. A few important distinctions as you're learning to build apps or as you are building apps. When you use the app wizard, which is the DX app wizard, you will have to either write it primarily in Bash or Python. You in turn can then use any executable you'd like, which gives you the ability to add any executable that you want to use onto UK Biobank. So let's talk about the general overview that we need to keep in mind as you are building apps and how to use them. So first, on your own computer, you will use DX run, either the name of the app or the applet, or you will use the applet in the UI. Once you've started that applet, it will make a request to the platform, which in turn makes a request for the worker. All of the files that you need to transfer to that worker will then need to be transferred. This includes your inputs that you have laid out. You'll then run your computation. After that, you will have an output file that you will then need to transfer from the worker onto the platform. And then you will get a response from the platform that the job is finished. I bring this up from the beginning for one really base reason, right? As you are thinking about how you're defining your inputs and how you're defining your outputs, this is the process that you keep in mind. Just so you know that you're not suddenly eliminating an intermediate or forgetting to add, your fast A file, something like that, right? You're remembering all of those pieces as you're building. All right, so next let's talk about the development cycle on the platform. We have, with your development cycle here, you will write the code locally on your computer using the DX app wizard. Okay. From there, you will use the command DX build, which will build the applet and upload it to the cloud. Once that applet has been built, you will use DX run to run it in the command line, or you can select the app within the UI. And you'll run that applet on the cloud. Now that I've given you as many of the overviews as I can here, let's talk about the details, right? So there are multiple ways that you can build applets. You can start with a binary resource and add it directly into the resources folder while you're building your applet. Examples of these are executables from GitHub. So these are the ones that are already packaged really well. These executables don't have a lot of other dependencies added to it. They're all bundled really well together. And so you just need to add whatever you're wanting to run into a folder for it to run. Then you have assets and Docker images. Assets are a DNA Nexus unique item, and then Docker images as well. These are for more complicated builds, such if you have a lot of dependencies that you have to worry about. The favorite here tends to be R when you're using something in R. This is when you'll need to use one of those two, so that way your executable will be able to run on your instance. And then finally, you can install executables as part of the applet build. So you can use your favorite package manager here to install these at runtime. Examples there are apt or cran. There are a few others that are already pre-built, and they are present within the Academy documentation that Brenton has listed earlier for you to view, okay? As I mentioned earlier, when you are using the DX App Wizard, your first bit of code should be written in Bash or Python. Again, you can then use any executable within that you'd like. Okay. Finally, the details on how to build each one of these is within the Academy website here. There is a building applet section, and there are examples of all of these for you. Okay. So when you're thinking about building your applet, there are many things that I have a list for you to think about. One of them is, how many dependencies do you have? Right? Do you know your inputs and your outputs? Do you have an output? As well as, do you want the intermediate files? Right? This is cloud computing. So if you do not care about those files, you do not have to have them as an output. Right? That's up to you. And then computationally, my main questions, right, as I'm starting to build these is, what size of the files do you have and are you intending to use? And is there a variance there? How much computational power is required to run that executable? And finally, what region are you using? Generally speaking, when when we're talking about computational power and files, this is going to be also part of up to your judgment. But an example that I have, right, is if you're using something like FastQC, it generally requires less computation than a read mapper due to the execution that's being performed. Also here, as we're talking about some of these options, you can start thinking about, within that computation, would it be easier for me to build it a certain way? Would it be easier for me to use a Docker image? Would it be easier for me to use an asset? Would it be easier for me to use a binary resources package built in? And finally, what are the requirements to build an applet? Once you've used the app wizard, you will have the following folder structure. So you have the name of the applet that you've given as your main folder, and then all of these are subdirectories or subfiles within here. You have your readmes, and you have a dxapp.json file. These are all of the pieces that you have built within the app wizard and all of the parameters you have set. That's here. Okay. You have a resources folder. This will be dependent on the build that you're using, but this is incredibly useful, particularly if you're using a binary resources. And finally, you have a source folder here, and this will have the name of the applet.sh. This is the required code that you'll need. If this is what will have to be written in Bash or Python. And you will have selected that when you're using DX App Wizard, and in turn, it will give you a template to be able to use, but you can style these however you'd like, as long as you're staying within Bash or Python. Okay. Finally, there is a test folder if you would like to use that as well. What is required at minimum is your JSON file and the source code. Okay. And just as an example, when you are building these applets, you can use dxget to be able to pull that applet and edit it. When you do that, it will form the directory. So here's that applet. And you see here that these are the two required pieces every time. You have your JSON file here, as well as the source, and then your code here. If you have a resources folder with how it's nested, you will have the resources. And then if you have a user bin folder, things like that in there, those will all be there as well. Resources, I did forget to say this, is whatever is in your resources folder will be downloaded onto your instance type at runtime. That is it for my overview. I did want to highlight a few of the resources for you all. So there is the tool library, which are public apps for you. There's some information here. You will be sent all of these links, so don't worry about that. There are tutorials here within the Academy documentation, as well as the UKB Introduction to App Building. You have your UKB-RAP documentation. And then another common question that comes up often is, how do I select an instance type? Those are here.
Well, that is awesome. And yeah, I mean, I think about two-thirds of you have probably built apps. You're probably wondering about how to select instance types. Also, a lot of people don't know about DXGet yet. So certainly in the sort of Q&A panel discussion, I'll be asking Emily a question or two about DXGet. That said, like I said, two-thirds of you have probably built apps or applets, or at least tried. Although most of you have been successful. And so what I'd like to turn over now, or I'd like to turn the floor now over to Xing Chen, who has built lots of apps, and he's really just getting there. And he's maintained sort of a list of gotchas, pieces of advice that he can help you with. And so without further ado, here is Xing presenting tips and tricks, really, for building apps.
Perfect. Yeah, thank you for coming. My name is Xing Chang. I'm a postdoc researcher in Oxford in the wearables group with Aidan Doherty. And yeah, so in my work, I was tasked with the trying to package some of the things we have so that we can use it in the RAP system and DNA Nexus system. And yeah, so let me start with clarifying that I'm not in DNA Nexus, but I'm sort of here as a giving us an outsider view of my experience trying to build an app in the UKB-RAP using DNA Nexus. And yeah, so just the list of gotchas that I collected while trying to do that. Yeah, so I'm not at all an expert, so just sharing my experience trying to build a very simple Python app. And so so let me just walk you through my experience with an example. So maybe it's a demo app where we try to read a CSV file. So it's a very common workflow. So it's an app where Python app where we try to read a CSV file. We try to run some processing on that using a very popular package such as pandas. We do some calculations and then we return a summary, maybe a JSON file, for example. And so that's sort of like the very typical script that you would probably run in your research. And so that might take that might be very straightforward Python script if you do it locally. But then the challenge that I had is how do I run this on the RAP system? And so, like Emily said, the DNA Nexus app wizard is a very fantastic tool. So I started from there. And when you use that wizard, which is very simple, it gives you all the instructions on what to do, what are the inputs that you need to have and outputs and the names and the metadata. Once you go through all of that, you get something like this. So a folder structure with some template files. And in this template file, the most important, perhaps the most important template file is the dxapp.json. That gives you the information, the configuration of the environment that will run your script. And so you have some metadata in there, like the name of the app, the version of the app, the input specifications and output specifications. And so here I told the wizard that I'm going to take a file and output another file, and also some optional input, which is n, a second input, which is going to tell the script how many rows of that CSV file I want. And so it's a very simple template. The first, let's say, gotcha that I found is that when I tried to build this, it threw me an error related to the region. And so when you walk through the wizard and then you try to build the app, it will say that your region has to match the region of the app has to match the region of your project DNA Nexus account. And so that's when I realize that you have to sort of dig into the JSON configuration file and change the line. Let me just get my annotator. Okay, so I think the mouse is not working. Okay, so there's this option here where it says regional options, and you have to change the region of that. And so for people based in the UK, that is very likely to be EU West 2 or 1. But yeah, that's one small detail. So when you want to build this, you have to specify the AWS region. So yeah, sorry. So when we build the app, the configuration region has to be the same as the project region. Otherwise, it won't build. So the other template file that it creates for you is this .py file under the source directory. It's usually named after the name of the app that you gave in the wizard setting. And so when we open that, I already modified this a little bit, but what you will have normally is a very simple template. And something I struggled with when I was trying to change this to fit the specific program that I was having in mind is the fact that the input file will not be the name of the file. So it will be this DNA Nexus link to the file, and that gives you a string, which is a file ID, and that file ID is actually the same that you get when you go to the browser and open the UK-B-RAP and go to your project and click one of the files. The file ID that shows up there, that's the file ID that you will get when you run the app. And if you want to get the real original name, original file name, there's an option which is, I think, is hidden there. So it's describe, it's .describe and then the name. And that's sort of a small detail, but it's sometimes important for certain applications where you need to know the extension so that you know whether it's a CSV file or JSON or Parquet or so on. So that's something also that took me some time to figure out. But yeah, basically the input to the program won't be the file name. It will be this file ID given by DNA Nexus. And so, but there is a way to recover the original file name. And so, yeah, and so like Emily said, we have to keep in mind this cloud mental model. So there's the DNA Nexus, what I call project space. So it's the space where with all your files, all your apps, and so on, the space that you see when you log into DNA Nexus. And then there's the job space. So DNA Nexus will spin out a completely new environment from scratch that is completely empty, let's say. And so you sort of have to download from the perspective of the job space. You have to download the file from the project space into the job space before it can actually run. And so this is sort of a pre-step towards the actual business code. And when you generate the files with the wizard, it will give you like a simple example of how that is done. But yeah, so that's something that to keep in mind. The other major problem that I have when trying to create the app is actually using third-party libraries. So let's say I want to read the CSV file with pandas, a very typical workflow in Python. And so where is pandas exactly? So if we try to do import pandas in that initial .py file, we will get an error because we haven't installed pandas anywhere at no time. And so this is the part where it took me some time to figure out. So we sort of have to first build a so-called DNA Nexus asset. And so that's something that is completely different to building an app. So the DNA Nexus asset is essentially a JSON file. And so you start with a JSON file with a configuration, with the OS that you want this to run, and the target OS, and then you give it a list of things you want to install. It can be Python packages, it can be CRAN packages, or app packages. And then once you do that, you build the asset. One thing to keep in mind is the version compatibility. So, for example, if you want to use Pandas 2.1, you have to make sure whether that's compatible with the Ubuntu version that you're running this on. And so if it's not compatible, it won't when you try to build the asset, it will fail. That can be, you can check that when you go to the relevant packages that you're trying to install, and you go to the change logs, or you go to the there will always have like a list of versions and compatibilities. So when you build the asset, you get a bunch of messages. You get something like this on the left. You will get an ID for that asset. So you then copy that ID, and then you go back to the DX app JSON. So this is the one that is generated by the wizard. And then inside that, you have to add asset depends, and then add the ID. And that's how you sort of install pandas, and then you make it available into your app, and then you can do import pandas. And then you have to rebuild the app so that you can actually import pandas. So that's sort of like the main things that were sort of difficult to find in the tutorials. And to summarize, this is the full script. So 2/3 of it is post and pre-processing to trying to deal with cloud, and then the actual business logic is 1/3 of it. Yeah, so I will make these examples available for anyone who wants to have a look. Yeah, that's it.
Wow, Xing, thank you very much, and I'm looking forward to having some like roundtable discussion on some of the things you were talking about. But also, I'd highly recommend people note this GitHub repo if you have any interest in wearables in general, and also just demo apps. Although there's a lot floating around out there, but Xing's group is really a world leader in thinking about wearables, and certainly on the UK Biobank. That said, our next talk will be given by Phil Greer. You've already received some of the GitHub repos and such that Phil Greer has put out there for the UKB-RAP. So he'll show you a lot of things in terms of sort of GWAS, as well as sort of building apps to sort of migrating over from HPC. I think that this is topic that people ask me every day, and I really appreciate Phil being here to speak about it.
Okay. It's nice to be here and talking to you about this topic. I've been working on the RAP since I think it came out before public beta. Something not everybody is going to know, not in the introduction. For about 10 years, I actually ran a small Beowulf cluster and train people on it. So I know a lot of ins and outs of doing high performance computing and working on that end of it. I am going to mainly talk to you about how to use the UKB-RAP and like step back from the app talks that we've actually already had and discuss how to use the UKB-RAP in more of a traditional HPC cluster or parallel cluster system. Move this. So first, some assumptions that, again, we've already talked about that in at the beginning. You know how to use a Unix command line interface. And by Unix, I'm talking about Linux and Mac. I think you can do all this stuff in like Cygwin on Windows, but I'm really not a Windows person. And hopefully most of you aren't either. But like if you are, I'm sorry. I'm not going to be able to like specifically talk to what you're going to be doing. Um, You've written at least one shell script. We're going to be mainly focused on Bash, but like any of the shell scripts, shell, C shell, T shell, TC shell, corn, whatever, as long as you've actually written something. You've used a batch scheduler for an HPC system before. If not, that's not a big deal. We're going to go over at least something about us, at least a Slurm job really quick. You have that UKB account and that you have DNA Nexus DX toolkit installed on your local machine. So all of this that we're going to be running is all going to be on your local machine. It's not going to be something that you're running in the cloud. Any job that you're going to do is going to be in the cloud, but you're going to be writing up scripts that are all going to be located on your local machine. So quickly, we're talking about like what are a job scheduler, and just talking about HPCs in general. So a job scheduler normally runs on the head node or the login node of an HPC cluster. The HPC cluster is built of a bunch of compute nodes, and that's where almost all of your jobs get done. And then it also has a file server and some shared disks, and I get a whole bunch of other things. Um, there are three types of jobs that you would run on a traditional on-prem HPC system. Distributed tasks, and that's probably the one that most people are familiar with, not most people are familiar with, but they're the ones that are popular. These are jobs that run across multiple nodes and require a high-speed interconnect in order to actually like get them to work well, because there's a lot of crosstalk between each one of the tasks that are located on each of the compute nodes. So high performance LinPack, the benchmark for supercomputers, is that type of job. It runs across all of the jobs and does work on every single one and then collects it all back at the end. They really tend to work poorly on cloud infrastructure because cloud infrastructure cares about reliability, or not necessarily performance, not network performance, but it cares about redundancy and making sure that enough resources are available. The next type is a perfectly parallel job, and that's the sort of job that runs like one compute, one memory, one system, and it doesn't talk to anything else. And you can run a whole lot of those jobs all at the same exact time. So there is no dependency on something that's happening in another job. That's the sort of thing that we're really going to be talking about here, and those work really, really well on cloud infrastructure. Finally, there's an interactive job, and an interactive job is the equivalent of actually like SSHing or opening up a shell on one of the compute nodes. And in fact, that's exactly what you're doing whenever you fire up a Jupyter notebook on DNA Nexus. You're creating an interactive job that fires up, as Emily talked about earlier, a cloud resource compute system, and you're then running interactively with it. You can actually, in the Jupyter Notebook, if you look at the very top of that Jupyter Notebook, open up a shell, and you can actually start using the command line on any one of those. You don't have to be contained inside that Jupyter Notebook. Next. So this actually is a Slurm job submission script. I don't know how many people you have seen this. I hope you have in the past, but we're gonna go over a little bit of it. So this is a bash script, but it has a whole bunch of interesting stuff at the very beginning of the bash script that is all related to the Slurm batch command. The very first S-batch line is the maximum wall time for the job that you're actually running. The next two lines are the number of nodes and the number of cores that you're going to be using. Next is like the job name and an email that the type of notification you want and when you want it out, when you want your notifications. Then there's typically on most clusters a program called module, and that actually loads information about the programs you're actually going to be running. And then finally, your actual software run command. And that's how this works. You actually then do an SBatch to install to run that, and you run this script, and it will then interact with that job scheduler to allocate the resources needed to run your job. You can run a whole bunch of jobs like this, or you can run that job that I talked about, the distributed job that would actually use like 5 nodes and all of the cores and all of the network interconnects. This is a DX run script. So this would actually be what you'll be running on from the RAP. So we don't have on the UKB-RAP a traditional job scheduler, but we do have a way to launch a program on a VM and allow it and give it information to run. For this one, we're using Swiss Army Knife, which is an app that DNX has built for you, for anybody to actually run. It has almost all of the common bioinformatics tools and genomics tools that you would actually want to use. BCF tools is on there, Plink 2, Plink 1, all of these different things are on there. The cool thing about this and how this whole entire thing is built, and I don't have a cursor on that, is actually, I guess you might see that cursor, that we can do many commands in this run Plink command. So I've actually got a run command that we're actually going to do, but it's actually doing 2 Plink commands back to back. So it's not just doing one thing. It's in a typical Unix fashion, running a command, getting a semicolon, which means that command is done. And after that one is completed, do this next command next on this same line. And so we're running actually 2 separate commands. Each one of these has a large number of variables that we actually define at the beginning, the imputed directory file, the data field, and all of these things are for where you're going to write data, where you're going to read data from, and information about the data you're using. So that most of that, at least the data field and the I information, are for a specific thing. So this is actually I is going to be chromosome 1. Data field is this UKB22828, which is one of the, well, I believe the imputed data code for the UK Biobank in the bulk generation file. And that is all used over and over again within the Plink file. The imputed file directory is actually used in the DX run command for the in files that we're actually using, so that it can find those files that it's going to copy into this virtual machine that you're going to build. The command, again, was before. The data file dir is actually the output directory. So that's the output directory where everything is actually going to write. So your destination, your project ID, and then the output directory. And so everything that is generated actually in this VM will get written back to this QC test directory. The other most important thing, and it's in white on this, is your instant type. So this is probably the question that was like asked the most on a regular basis in the community forum is what instance type am I supposed to actually go run on. There's a lot of things that aren't on here that you would find normally in that Slurm script. The things that you're missing is like the wall time. You don't really need that. It's just going to run to completion, and it doesn't matter. Your email notifications, that is all handled on the UKB-RAP portal. You can turn them on and off and decide when you're going to get emails in that web portal. And then that module section doesn't really matter. That is all controlled by the app that you're actually going to start. So let's actually talk more about this, the actual instance type. If I actually hit these two little brief and yes, those are just commands so that if it ran into an interactive thing where it was asking you, do you want to do this? Yes, no, it will automatically accept a yes. So that you don't have to actually like accidentally hang it while it's waiting for a yes or a no response. So let's talk about the next slide. So your virtual node. So we have DNA Nexus has a UK Biobank rate card with all of the different rates and different nodes that you can possibly use. A lot of these things are extremely important for what you would do to try to figure out what sort of node that you're actually going to run on. I'll show you some instances on that rate card in the next slide. But different things that change constantly is the number of virtual CPUs that you actually have on your VM node. The more CPUs, the more RAM is going to be available. How much RAM is available, that's actually the second one. And then the storage for your local files. One of the things that I really find is that for storage, you really want about 4 to 5 times the file size that you're reading because you don't know really how big any of the outputs are. So if you're dealing with like chromosome 1 on the imputed genotype file, you're talking like, many hundreds of gigs. Whenever you manipulate that file, you're going to create another copy of that with many hundreds of gigs. And you don't want to actually go ahead and run all of this and then run out of disk storage space. So pick something that's about 4 or 5 times larger than the file inputs that you're actually dealing with. It could be that something that you're going to be doing is going to create a really, really small output file. But if you don't know how big it's going to be, it's better to just pick a really large file. Something that's on here is SSD versus hard drives. And the things that you have to remember for SSD versus hard drives is that SSDs have really high input-output operations per second. Hard drives don't because they're spinning platters. But this is all cloud instance, so these are all going to be running on RAIDs systems. They're going to be still pretty fast. They're not going to be like the laptop hard drive from your 2004, 2012 laptop. You're actually going to still be getting like pretty decent performance on the hard drives.
Bill,
So it doesn't really
everything
matter.
you're saying is super relevant, but if I could ask you to wrap it up in the next 3 or 4 minutes, that would be awesome.
Okay. I'll try to go through this really, really fast. So it doesn't really matter whether you pick the SSD or HD for most of the time. I'll skip the next couple, but like spot versus instance pricing. Spot works great for anything that is under 2 hours long, and it will continually work really, really wonderful. And you can use that cheapest instance as much as possible. If you're going to be running something for more than 2 hours, and here I would test it on one chromosome rather than submitting all the jobs at once, you want to do it on the standard instance so that it will actually go ahead and run fully to completion. So one of the things that we have that wonderful looping with that we have the DX run command. So one of the awesome things about that is that you, for most tasks that you're going to be doing, a GWAS, frequency calculations, Hardy-Weinberg equilibrium, any of those things that you're actually going to be doing, they're going to be independent. You don't have to have all of the data read into a single instance. If you're doing something like epistasis, that's different. You might actually need all of your data together in order to actually like get some sort of information collected across all chromosomes to look at something like that. So this is the part in Bash where you start looking at loops, like for loops. This first one that we're actually talking about is just looping over the 22 chromosomal, the autosomes. You do a for I, again, that was in that previous one that we were looking at. I is now going to be from 1 to 22, and it's going to run 22 separate DX run commands for you. If you want to include X, you might actually have to set them all up separately so that it can actually include X onto it. For whole genome sequencing is available for everybody, and it's in different data chunks. So your different data chunks, I think on the first initial whole genome sequencer release, they were in 50 KB kilobase data chunks. The new one, the larger one, is in 20 KB data chunks. You can actually pick a set of chunks and actually go ahead and run through all of it and loop through all of it and manipulate each chunk separately. So this is the DX run script that I actually showed before, but now we've actually set it up so that it is actually looping over I. So it's looping over chromosome 1 through 22. And this is, again, the thing that is going to create 22 jobs that are all going to run at the same time. 1, 2, 3, and 5 are going to be the longest, and they're the largest chromosomes. They will take way longer than chromosome 20, 21, and 22. But most of these will run, this actual program, I think, was running in like less than 11 minutes for almost all of them. So this sort of thing can actually run really, really fast on this memory size and will work wonderful for you. A different way of doing that loop is a while loop. So if you were doing something that didn't have that continuous factor, so 1 through 22, if you had a list of MRIs that you wanted to normalize, segment, sample, something or other, if you had a whole bunch of BAM or CRAM files that you actually wanted to actually do a BWM alignment, you can actually have each one of these run separately. And you would do a list of all of those. I actually had a similar version question request to list all of the VCF files from whole exome or something like that. I have a script like that on my GitHub repository so that you can actually get all of that information, put it into a file, and then read each line. Each line has a name for each line, and you actually will just loop through that whole entire file and run a DX run command. So you can actually have hundreds or thousands really of jobs. Other things that you can do with this is Monte Carlo simulations, where random Monte Carlo simulation on Hardy-Weinberg equilibrium resampling inside of in the UK Biobank. That's all on my GitHub repository. I was looking at extracting all of the regions, all of the whole genome sequence data for a specific gene. Read this slide at your leisure if these slides are actually all getting shared. I actually built this code to actually do that, but I'm not really going to go into that one further. And we can talk about, I put at the very end all of the references I've talked about, so that you can think about all these things. Really, the sky's the limit. You can do so much with the DX run command from your local command line. I have barely scratched the surface with the things that I want to do. So thank you all for
Awesome.
your time.
Phil, thank you so much. All right, we have about 7 minutes for questions, and we have a lot of material to get to. So I'm very excited about that. So if I could ask all the panelists to come back. And I actually want to start with an audience question, which I don't usually do. And this question is, if I want to create an applet starting off a whittle pipeline, what's the best way to do that? And maybe we'll start with Emily on that one.
So if you have a whittle pipeline, you can use an importer. That is already a tool in the tool library for you all. If Brenton, you could send the link out to it. That's how you would import an existing whittle pipeline. If you are DIYing one directly onto the platform, there are some resources as well within the Academy documentation, and you'll use the DX command, DX compiler, to be able to import that DIY pipeline as well.
Awesome. Yeah, the DX compiler is up there on GitHub and easy to use. Question for Phil then. I think for a lot of people it's really tempting just to use like an array of large Jupyter notebooks. For even GWAS or even iterative GWAS. Can you sort of talk about from your perspective why you think that may not be really the most efficient way to run things?
I think there's two answers to that. I mean, or there's two reasons for that. I think the Jupyter notebooks are all on standard and not spot. And they're going to be a lot more expensive in general because they're on standard specific. They're actually like the highest set. Most of these GWAS will actually run really, really, really fast if you run them all independently on a per chromosome basis. And I'm actually talking about using all, hundreds of thousands of people. They're really, really super, super quick to actually run and will run each chromosome in well under a half an hour. If you're actually doing it that way. So I think setting up the scripts and depending on your tools, because you have a lot of different tools that you can use. You can use Plink, you can use Regini, you can use all of these different other steps that you to actually run your GWAS. You can do it on so many different data sets, whole exome, imputed, possibly even the GWAS, the full whole genome later. But I haven't really actually gotten to that one yet. I think it's better and faster to do it separately on each of these scripts and know how to combine the data at the back end than it is to actually try to do it interactively in a Jupyter notebook ever.
Great. Thanks, Phil. There's a great question online, but I wanted to ask Shing something I'm personally very curious about real quick. Shing, there's a variety of expertise in your lab. You have all the way from epidemiologists to people who are doing really sort of hardcore accelerometry stuff. How does peer-to-peer learning work there? Like how do different people work together to help each other learn these different techniques within your lab?
Yeah, so I think in the way Aiden has structured the group is let everyone double down on their expertise. So if people who know GWAS, they just do GWAS. People who know EPI, they just do EPI, and so on. I think we try to obviously make it so that the unit of work that can be shared between the 2 or 3 people involved is very clear. And then so for example in my work I'm very much focused on phenotyping, so deriving phenotypes from sensor data, motion sensor data, or ECG or accelerometers. And once I get the phenotypes, I sort of delegate the rest of the analysis to the more epidemiology focused people or the GWAS people. So that's how we manage in our group. Yeah, and so in the case of UKB-RAP, I think the good thing is that there's a clear separation between using Jupyter notebooks and using apps and applets. So for the phenotyping, we would run apps, and then once we have all the results and collate them, then we can do analysis on the notebooks. So I think that distinction is pretty clear, at least in our group. Yeah.
Cool. Thanks, Shing. All right. Ed has a great question, which says, do you have any advice on how to process many chunked VCF files in parallel? Tried doing this with workflows, but doing this across hundreds of thousands of jobs, once we hit the 100-job limit, causing the workflow to hang when there were no workers free. So there are a couple of tips and tricks for chunking, and they're very important. That said, I will take the moderator's perspective and start off here by reminding you that if you're working on a specific project and you need a few more workers, you can write an email to UKB support at dnanexus.com explaining what you're trying to do and your request, and you can get bumped up in terms of the number of worker nodes you have. That said, I'd like to go to maybe Phil for a very short answer, and then Emily on ways you strategize chunking VCF files.
So for how I strategize chunking VCF files, I've mainly been using the chunks as published and building loops across that. I am aware that there are those issues. I think I was seeing somebody has started to write a tutorial, but it's an older one on running Shape-It to phase all of the whole genome sequencing data in UK Biobank. And they do this sort of this DX run with loops over chromosome over chunks, so that they could actually get that fully phased data. And that's at the Shape-It 5 group. That would be how I would do it. I don't think, I mean, that is obviously again, that issue with running into the ceiling with how many jobs can run at the same time. But that's what I would do.
Cool. Emily?
So interestingly enough, that is also my answer, is to do it by the chromosomal amount. But if you, as Ben mentioned, if you have concerns about that job limit, you can, ask for those resources as well if needed.
Yeah, exactly. I mean, the big deal is that we can't crash our part of the London data center. And if you tried requesting increased resources right when we were releasing the 500K, we absolutely could not process resource increases because there was a huge spike in usage. But now that should be sort of more or less doable. That said, we are down to -2 minutes left. So I would like to ask all the panelists one question, which I ask at most of these roundtables, which is, what is the coolest paper you have seen on come out of the UKB lately? And I think that dovetails well with another audience question. So, Shing, do you want to take a shot at that?
Well, I'm obviously biased, but I will say the paper that one of my colleagues worked on. Yeah, he managed to train a deep learning model on terabytes of data. So the accelerometer data set. So this is the self-supervised learning for activity recognition work. Yeah, so it's sort of like the first foundation model for motion sensor data. Yeah, just want to, obviously I'm biased, so yeah. So.
No, I mean, I think we're all fascinated by motion sensor data and how that plays into cardiovascular health. So that's super cool. Phil?
I think I was excited the most about the Olink data. But I have, again, not had enough time to actually like dig too much into it. I think that was really cool. There's so much that can be done with the data. And I look forward to seeing not just the Olink data, but just data in general on the UK Biobank, of looking at interesting ways of using it that people haven't thought of before. Whether that be simulations, whether that be like questioning, just general ways of actually like QC work or things like that. I'm really interested in that sort of thing.
Awesome. Emily, last word.
I particularly like the longitudinal studies that are present, including with the COVID studies as well.
Awesome. Well, yeah, you heard it here first. When we ran a scientific conference recently, half of our participants said they would like to see longitudinal proteomics. So hopefully that'll happen with the UK Biobank and other data sets. With that, this is a wrap. We have a few more webinars coming up this year. And then we will be back strong in January talking about lots of other topics, and we're going to have a scientific topic calendar for the first half of 2025. So stay tuned for that. Thank you all for coming. As always, if you have questions, ping me your support. And there's a survey that Brenton just put in the chat. So if you have time, hop on that, and then we'll be out of here. Also, we'd love to hear your ideas for other webinars, roundtables, scientific topics, etc., etc. Finally, I'd like to thank our panelists one more time. And with that, have a great rest of the day. And we're heading into the weekend, so have a great weekend, folks.